Good afternoon. Dr. Grief and myself are happy to introduce this section, number 5, New Science from Young Talent. And so this, I guess, the resident and the trainee session, and they will present some exciting results. So we'll start with Meghan De Meo from Jonathan Spicer Lab on the KRAS inhibitor to overcome neutrophil-mediated immunosuppression. And then we'll have time for one question, hopefully, after your talk.
Thank you. Thank you very much for this opportunity to present. I'm excited to share my project with you. As we all know, lung cancer is the leading cause of cancer-related death because often they're diagnosed at late stages. We also know that lung cancer standard of care is evolving with immunotherapies, targeted therapies, combination therapies. However, the rate of response is still pretty low. Immune compartments in the tumor environment are very important in terms of patient prognosis and response to anti-cancer therapy. Specifically, we see that when there are high amounts of neutrophil infiltration in the tumor, they're associated with the worst prognosis and poor response to anti-cancer therapy. And so neutrophils are recruited mainly through the CXCR2 receptor, which is the main chemotactic receptor, and this is mediated via CXCR2 ligands. And so we will look at this axis in the context of lung cancer. We also know that lung cancer is divided into 2 main histological subtypes, non-small cell lung cancer accounting for the majority of cases. And within this subtype, there are 3 more subtypes: adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. And taking a closer look at these 3 subtypes, we see with human publicly available bulk samples that neutrophil infiltration in these tumors is associated with the worst survival in lung adenocarcinoma. We also see that high expression of most of these known CXCR2 ligands is associated with poor survival, specifically in lung adenocarcinoma, which is why we will focus the presentation on lung adenocarcinoma. There's an overwhelming amount of evidence suggesting neutrophils' role in cancer progression and metastasis. And as well, our labs were the first to show that NETs, neutrophil extracellular traps, first known for their protective role against foreign pathogens, trap circulating tumor cells and promote distant sites of metastasis. Neutrophils are also known to suppress cytotoxic T cell function and promote exhaustion of these cells. Immune checkpoint inhibition, so anti-PD-1, PD-1 therapy was discovered to counteract this mechanism. However, response is still quite low. So this leads us to our question: how do we select patients that would benefit from neutrophil recruitment therapy? And so when patients come to the clinic, they're routinely screened for driver mutation status. We know that KRAS is among the most prevalent driver mutation, and this is also upstream of some potent CXCR2 ligands. And so for this reason, we wanted to see whether we can use driver mutation status as a surrogate marker of neutrophil infiltration in a patient's tumor. We first looked at the Cancer Cell Line Encyclopedia, where we found that human lung adenocarcinoma cell lines had the highest expression of CXCR2 ligands. This led us to our hypothesis that KRAS-mutated lung adenocarcinoma might have the highest neutrophil recruitment, and that combining neutrophil recruitment inhibition in this cohort would be beneficial for increasing anti-PD-1 therapy efficacy. And so we do this through 2 major methods, and both novel microfluidic devices. This is our 2D device, where we have 6 outer wells and 1 main channel. In the outer wells, we load tumor cells. We allow them to secrete cytokines for a period of 24 hours, and then we load the neutrophils in the main channel and allow them to migrate. We have a 3D device which mimics immune cell migration through the blood cell, through the extracellular matrix, and finally into the tumor. And we model this with a membrane, a collagen layer, and finally spheroids. So we first found that neutrophil recruitment was highest in all of our KRAS mutated cell lines. We then found in a co-culture assay that KRAS mutated conditioned media induced the most amount of netosis, specifically from KRAS mutated lung adenocarcinoma cells. We echo these findings in our 3D device, where we see the highest netosis burden, shown in green, in our KRAS mutated spheroids. So now we want to know whether we can use neutrophil recruitment inhibition to improve anti-PD-1 therapy in these patients. We isolated CD8-positive T cells and allowed them to activate for a period of 48 hours. On day 1, we seeded the cancer cells. We allowed them to aggregate and secrete cytokines for a period of 24 hours. And then on day 2, we add neutrophils. 4 hours later, we add CD8-positive T cells. And 4 hours later, we intervene with anti-PD-1 therapy. And we do this to mimic the innate versus the adaptive immune system. And so we did these experiments following 3 guiding questions. And the first is we wanted to understand what role neutrophils play on T cells in these systems. So we first see that when neutrophils are in the system, they actually suppress T cell infiltration into the spheroid, as seen in blue. As well, we see that tumor cell killing, represented in red, is much less when neutrophils are involved in the system. Then we want to understand what the effect of neutrophils were on anti-PD-1 therapy efficacy. And we see that when neutrophils are in the system, there is much less tumor cell death. We then wanted to know whether we can use a neutrophil recruitment inhibitor to increase anti-PD-1 therapy in these patients. And so as a neutrophil recruitment inhibitor, we wanted to try KRAS G12C inhibition, since this is upstream of potent CXCR2 ligand expression. To confirm this, what we first did was we performed an ELISA of 96 chemokines, and we compared the secretion of cells that were KRAS G12C mutated versus a control KRAS G12V. We did this with and without the KRAS G12C inhibitor, and we see differences when we had the inhibitor present in these cells. Specifically in the neutrophil recruiting chemokines, if we take a closer look to the 2 most potent CXCR2 ligands, we see that this inhibitor almost mitigated the expression of these cytokines, but didn't really have any effect on our control KRAS G12V cell line. So then we wanted to understand, would this impact neutrophil recruitment in our devices? And in our 2D device, we see that the KRAS G12C inhibitor reduced neutrophil recruitment. As well, we see this in our 3D device. So finally, we added a KRAS G12C inhibitor to the system, and we see that adding this inhibitor overcame the neutrophil-mediated immunosuppression we were seeing and resensitized the tumor, which resulted in an increased amount of tumor cell death. And so to conclude, we propose that neutrophil recruitment inhibition could be used to benefit KRAS-mutated patients to augment anti-PD-1 therapy. And this is especially important since these patients have currently a limited amount of clinically approved targeted therapies available to them. In light of all of the ctDNA work being presented yesterday, we want to share our recently published findings where we were able to detect KRAS G12C ctDNA in patients in a very cheap and very quick manner using ddPCR. So taken together, we propose that targeting the genetic and mechanistic link between neutrophil infiltration, KRAS mutation, and detection can improve patient selection and efficacy in anti-PD-1 therapy. Thank you very much.
So maybe I can start the first question. So in the preclinical model of the KRAS development, they also show upregulation in mice of CD8 when the patient or the mice receive KRAS inhibitor. And in your study in the human post-KRAS inhibitor, was there a change in the neutrophil in the biopsy post-KRAS inhibition?
Would you be able to repeat them?
Yes, so you show a study or in the trials that are ongoing with KRAS inhibitor in the post-biopsy specimen. Do you know if there is a change in the immune contexture in the human?
We haven't done this in humans. We'd like the direction to go in that way. However, these are only 3D devices that we use with human cell lines for now.
Thank you.
Great, thank you so much, Dr. De Meo.